Okay, have you ever really thought about how some viruses can infect you once and then just stay with you for your entire life? It's an incredible biological mystery, right? These things are masters of disguise. They hide out silently, right inside our own cells, sometimes for decades, only to pop back up when we least expect it. So today, we're going to go undercover and expose the secrets of two of the most successful viral stowaways out there.
All right, here's our game plan. First, we'll meet the culprits. Then, we'll track them to their hideout, deep inside our nervous system. After that, we're opening up 2 different case files to crack the code each virus uses to stay quiet. And finally, we'll get into why all of this matters so much.
So, let's meet our two main subjects. They're both part of the alphaherpes virus family. One is Herpes Simplex virus 1, you probably know it as HSV1. The other is Varicella-Zoster virus, or VZV. And look, these aren't some rare, obscure viruses. They are incredibly common, and they have absolutely perfected the art of living with us for the long haul. And just to give you a sense of scale here, check out this number. Over 90%. That's how much of the entire human population is latently infected with Varicella-Zoster virus. Think about that. It's hiding out in the nervous systems of billions of people as we speak.
So HSV1, you probably know this one as the virus that causes cold sores, and it's hiding in about 80% of adults right here in the US. Then there's VZV. This one's even more widespread. It's the one that gives you chickenpox when you're a kid. But here's the kicker. It doesn't actually leave your body when the spots go away. Oh no. It just goes dormant, it waits, and then it can reactivate years, even decades later, to cause that really painful condition we call shingles.
So the big question is, how on earth do these viruses find such a perfect, lifelong hiding spot? Well, they take this incredible journey, starting from where they first infected you, say your skin or mouth, and travel deep into the protected territory of our nervous system.
It all happens in about 4 key steps. First, you get that primary infection. You know, your first cold sore or that bout of chickenpox. From there, the virus actually invades the very tips of your sensory nerves. Then, it travels up those nerve fibers, kind of like a train on a track, all the way to the main nerve cell body, which sits in a little cluster called a ganglion. And once it's safely inside that neuron, it shuts everything down and goes dormant. It's established latency.
See how they literally hide inside our nerve cells. For HSV1, a favorite spot is the trigeminal ganglia. That's a nerve bundle up near your brain. VZ, well, it's a bit more widespread. It sets up shop in ganglia all along your entire nervous system. And once it's inside the neuron's nucleus, the viral DNA just chills out as a little circle of DNA called an episome. It's totally separate from our own chromosomes. And because it stops making any new virus particles, it becomes completely invisible to our immune system. The neuron becomes the perfect safe house.
Okay, but think about this for a second. A virus is basically a replication machine. That's its whole purpose. So how does it force itself to just be quiet for years on end? Let's open up our first case file on HSV1 and find the molecular switch it uses to stay so silent.
This was the huge mystery for scientists for a long time. I mean, it's one thing for the virus to get inside a neuron, but it's something else entirely for it to actively turn off its own powerful replication genes, the very programming that tells it to make more of itself. So, what tells the factory to just shut down production?
Well, the answer is a fascinating molecule called the latency-associated transcript, or just LAT for short. Now, most genes are blueprints for proteins, right? Not LAT. It's what's called a non-coding RNA. So during latency, when all the other viral genes are dead silent, this one is being pumped out like crazy. Its job isn't to build anything. Its job is to be the manager, to regulate everything else. A huge piece of this puzzle came from a 2016 study published in PLOS Pathogens. It was from a team led by Michael P. Nickel, and their research gave us some of the first really solid evidence of what LAT was actually doing inside those sleeping neurons. And what they found was pretty clear. Basically, their data showed that the LAT RNA's main job is to shut down the virus's other genes. It's the key that keeps the virus locked in that silent state, which is crucial for stopping it from reactivating too often and for keeping that hidden reservoir of infected neurons around for a lifetime. So, to put it simply, think of LAT as the virus's own internal security guard. Its job is to silence all the go signals for replication. It actively stops the virus from waking up. This makes sure the neuron it's hiding in stays alive and well, which preserves the virus's safe house. It's even involved in what's called epigenetic regulation, basically adding chemical do not disturb signs to the viral DNA, locking it in a super deep sleep.
Okay, so that's HSV1. It uses LAT. For years, scientists figured, hey, its cousin, VZV, probably does something similar. But man, finding it was way, way harder. The trail went cold for a long time. So now, let's open up that second case file.
And then, finally, a breakthrough. The discovery of the VZV latency-associated transcript, or VLT. And just like they saw with HSV1, researchers found this one specific viral transcript that was always being made in those sleeping VZV-infected neurons, even when everything else was shut off.
The mystery was officially cracked in a landmark paper from 2018, this one in Nature Communications, by Daniel Depledge and his team. What they did was brilliant. They used some really advanced sequencing technology on human trigeminal ganglia, which they got at autopsy, and they were finally able to pin down and describe VZ's secret weapon.
And here's the smoking gun. What their paper showed was that one specific version of VLT is the main one you find during latency, and it has a very precise target. It shuts down a viral gene called OFR61. Now, OFR61 is like the master switch. It's the key that turns on the entire replication process for the virus. Without it, nothing happens. The way VLT works is really clever. It's antisense to that ORF61 gene. That just means its genetic sequence is a perfect mirror image, so it can stick directly to the ORF61 message and physically block it from being read. But there's a twist. Unlike HSV1, which goes totally silent, VZV keeps another gene called ORF63 simmering on low. We're still figuring out why, but it suggests VZV uses a slightly different strategy. Maybe it stays a little more alert in its hiding spot.
All right, so now we've got the dirt on both our suspects, LAT and VLT. Let's bring the two case files together and put them side by side to see exactly how their strategies compare.
So here's the bottom line. Both viruses use a special transcript to stay dormant, but their playbooks are totally different. HSV1's LAT is like a blanket suppression. It enforces this deep, widespread silence using those epigenetic changes. VZ's VLT, on the other hand, is more like a surgical strike. It uses that antisense trick to take out one single critical gene, ORF61. And this difference in strategy might actually be why they cause different diseases. Maybe HSV1's deep silence leads to those localized cold sores, while VZ's more alert state in ganglia all over the body is what leads to the widespread painful rash of shingles. So you've got two different viruses with one shared goal, but they came up with two totally different playbooks. It's a fantastic example of what biologists call convergent evolution. They both face the exact same problem. How do you hide inside a human neuron for life? And they both came up with a similar type of solution, a special transcript, but the actual mechanics are completely different. Okay, so we've been deep in the molecular weaves here, but why does any of this matter? Why spend so much time cracking the codes of these sleeping viruses? What's the real-world takeaway?
Well, it's because understanding these master switches, LAT and VLT, is everything. If we know exactly how the virus forces itself to stay quiet, we could potentially design drugs that help it do its job better. Imagine a medicine that just reinforces that latency, locking the virus away for good. This could mean preventing agonizing shingles, stopping viral infections that cause blindness, or even preventing deadly encephalitis. It really opens the door to a whole new way of thinking about antiviral treatments. And hey, if you want to take more deep dives into virology and pathology like this, you should for sure check out the Master Oral Path channel on YouTube. It's a great resource.
And that really leaves us with a final question to chew on. These viruses are ancient. They've evolved with us for thousands and thousands of years. They have perfected the art of survival inside us in ways we're only now starting to get. So, as we learn more about aging and immunity, you have to wonder, what other lessons will we learn from these, lifelong companions of ours?